One topic that we'll be looking today in biochemistry is the glycolysis, a very fundamental process, most probably the first ever energy producing process that was evolved during the course of evolution. Glycolysis involves the breakdown of glucose. So that's what the name indicates, glucose breakdown or glucose breakdown. We can also say this as a partial oxidation of glucose. So it's not complete oxidation, because complete oxidation means we should get carbon dioxide and water. It's only partial oxidation where a glucose molecule which has 6 carbons is oxidized to 2 molecules of pyruvate. So you don't have any carbon loss. But during this, 2 NAD molecules will be reduced to NADH molecule. So this is basically the oxidation process. It also provides energy, a net energy of 2 ATP molecules. This we call substrate level phosphorylation. And most probably this is the first ever kind of ATP synthesis that is evolved. In a substrate level phosphorylation, a high energy compound donates its energy to ADP and PA joining to form ATP. The pathway was elucidated by Emden, Meyerhoff and Parnas. That's why it is often called as EMP pathway. So Emden, Meyerhoff and Parnas, they made the elucidation of the pathway. They identified the various kinds of intermediates which are present and gave the sequence of events during the glycolysis. This is the anaerobic phase of respiration. Which means there is no requirement for oxygen. And remember, even in the entire glucose metabolism, the oxygen requirement comes only in the case of the electron transport chain. Until then, you have substrate level phosphorylations producing the ATP molecules. The pathway occurs in prokaryotes and eukaryotes. It occurs in anaerobes, occurs in aerobes. It occurs in unicellular organisms. It occurs in metazoans or multicellular organisms. So it doesn't matter where you go, which kingdom you see or which domain you look at it. Everywhere we have glycolysis as the basic fundamental mechanism for energy production. This is an important aspect. It is partial oxidation of glucose, where glucose is oxidized to alpha keto acid, which is pyruvate. There is no oxygen requirement. It produces NADH molecules that can be taken for electron transport. You can generate further energy from NADH molecules. Now when you take the glycolysis, there are certain important points that we have to remember. The first one is, there are 3 enzymes in glycolysis that catalyze irreversible reactions. So anyway, that irreversible reaction will be under physiological conditions. Thermodynamically, every reaction which is catalyzed should be reversible. But we have 3 enzymes that catalyze physiologically irreversible reactions. The first enzyme is hexokinase or glucokinase, depending on the tissue. Hexokinase in most of the tissue, glucokinase sometimes in the liver, glucokinase in the beta cells of pancreas. They will do phosphorylation of glucose to generate activated form of glucose. So that is the reaction where glucose is converted to glucose 6-phosphate. Remember, the ATP that is used for this reaction is magnesium complexed ATP. All hexokinases, they require magnesium complexed ATP as the reactants. So that ATP is 4 minus, ADP is 3 minus. That's the first reaction. The second reaction is phosphofructokinase or PFK reaction. So this enzyme converts fructose 6-phosphate to fructose 1,6-bisphosphate. Uses ATP for exchanging that phosphorus. This reaction is the rate limiting reaction of the pathway. So rate limiting reactions we show with thick arrows. So this is the rate limiting step or committed step or regulatory step. Doesn't matter what name it indicates. This is a step where the glycolytic carbon exchange, the glycolytic carbon movement will be controlled towards the pyruvic acid. The fructose for phosphofructokinase, the enzyme which is catalyzing it, is a allosteric enzyme. An allosteric enzyme is nothing but multi-subunit enzyme having a distinct site. So it has distinct regulator binding site, and that regulator changes whether the enzyme becomes active or enzyme becomes inactive. This allosteric enzyme has binding sites for substrate ATP. Remember, the reaction uses ATP as substrate. It has binding site for regulatory ATP. So this regulatory ATP binding keeps the enzyme in the inactive state. Then you have fructose 6-phosphate binding site. Then you have fructose 2,6-bisphosphate binding site. This compound, fructose 2,6-bisphosphate, is the major allosteric regulator of phosphofructokinase. It increases activity of PFK. So when you have high concentration of fructose 6-phosphate, some fructose 6-phosphate can be converted to fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate now stimulates phosphofructokinase. Fructose will be rapidly converted to fructose 1,6-bisphosphate. And then there is a binding site for AMP. So the allosteric regulatory phosphofructokinase consists of 5 distinct binding sites: substrate binding site, regulatory binding sites, fructose 6-phosphate binding site, fructose 2,6-phosphate binding site, AMP site. This will be activating, this will also activate. So they are allosteric activators. Fructose 6-phosphate is anyway substrate. There is also substrate ATP. So we can give functions for all of them. Now the one that we will be looking at it is, what is the role of second ATP? What role it will be playing, because already you have a substrate ATP. Remember, glycolysis is something that produces that proceeds for energy production. Now you can have situations where the cell has so low ATP so that it can be used for ATP generation, or you might have case where there is a high ATP. In this case, you don't require phosphofructose kinase to be active, because you don't want further glycolysis in the cell. So that's where the importance comes. When ATP is low, ATP concentration is low, ATP binds only to substrate binding site. Because of that low level of ATP, ATP cannot bind to regulatory site. Since ATP is not bound to the regulatory site, the fructose 6-phosphate will be phosphorylated and you have continuation of glycolysis. At the same time, when you have a higher concentration of ATP, now ATP binds to substrate binding site as well as to the regulatory binding site. Now the binding is not limited to substrate binding site. Once ATP binds to regulatory site, it inactivates phosphofructokinase. That is how high levels of ATP in the cytoplasm act as inhibitors of glycolysis, because at high ATP concentration, ATP not only binds to the substrate binding site but also binds to the regulatory binding site, and as it binds to regulatory binding site, it will be getting a conformation of phosphofructokinase, which is inactive form. So typically we have two distinct forms of any regulatory allosteric enzyme. One form we call it as R form, this will be active. Another form we call it as T form, this is inactive. R stands for relaxed, T stands for tensed. So if AMP binds, it stabilizes this. If ATP binds, it stabilizes this. So AMP is activating, ATP is inhibiting phosphofructokinase. So that is the second enzyme which catalyzes irreversible reaction. Then the third enzyme is pyruvate kinase. So this enzyme is the one which converts the high energy phosphoenol pyruvate into pyruvate, producing ATP, requiring potassium. So those are the three enzymes that catalyze irreversible reactions in the glycolysis. Hexokinase or glucokinase, which catalyze the first reaction, that is phosphorylation of glucose. Phosphofructokinase, which catalyzes the regulatory activity of the glycolysis. Pyruvate kinase catalyzes the last substrate level phosphorylation reaction to produce pyruvate. Then there is only one oxidation step in the entire glycolysis. So this oxidation step is catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. So that will be the enzyme which will be making the oxidation. It is removing the hydrogen, so it is making the oxidation. This enzyme is regulated by inorganic phosphate. In anaerobic organisms, this step is one of the regulatory steps. The reason for this is, in case of anaerobic organisms, NADH cannot be subjected to oxidation by electron transport. So NADH has to come back and it should form NAD+. NAD+ is needed for glyceraldehyde 3-phosphate dehydrogenase. So we have the reaction glyceraldehyde 3-phosphate plus inorganic phosphate plus NAD plus gives rise to 1,3 bisphosphoglycerate plus NADH. So this is reduced compound, this is oxidized compound, and the reaction proceeds if you have NAD available, inorganic phosphate available. So in case of anaerobic organisms, they should rapidly regenerate NAD plus if they want to continue the glycolysis reaction. If there is a decrease in the NAD plus concentration, then glycolysis will not proceed. This enzyme is a thiol group containing enzyme. Thiol group means you have cysteine at the active site. And any group or any enzyme that contains thiol group can be inhibited by iodoacetate. Iodoacetate, iodoacetamide, mercury ions, all these will inhibit glyceraldehyde 3-phosphate dehydrogenase activity. Then we have two substrate level phosphorylation reactions. To produce ATP. So one reaction is catalyzed by phosphoglycerate kinase. So this enzyme will convert 1,3-bisphosphoglycerate to 3-phosphoglycerate with the formation of ATP. So this is your energy molecule. This is high energy compound providing its energy to ADP, making ATP. The second reaction is pyruvate kinase reaction. So that will be the terminal reaction already we got, because this is catalyzed by physiologically irreversible enzyme. It converts phosphoenol pyruvate into pyruvate. So again producing ATP. So now we got enzymes that catalyze irreversible reaction, enzyme that catalyzes oxidation reaction, then enzymes that catalyze substrate level phosphorylation. Then we have one enzyme that is useful in clinical biochemistry. That enzyme is the enolase enzyme. The reason is this enzyme is inhibited by fluoride ions. So when blood samples are collected for the glucose estimation, usually a small amount of fluoride is added. The fluoride will inhibit the enolase reaction, that is the conversion of 2-phosphoglycerate into phosphoenol pyruvate. As this reaction is blocked, the glycolysis will be blocked. In other words, once you collect the blood for glucose estimation, more or less the same amount of glucose remains until you go for estimation of the glucose. That's why that a small pinch of fluoride is added. Then if you look at the pathway, it starts with glucose. So we'll take one molecule of glucose. One molecule means 6 carbon, always balance with your carbons. This is energy utilization step, where ATP is used to produce ADP. This is catalyzed by either hexokinase enzyme or glucokinase enzyme. Mostly hexokinase, but as I said, sometimes in the liver and always in the pancreatic beta cells, glucokinase works. It produces activated form of glucose, which is glucose 6-phosphate. The carbons remain same because the only thing that happened is phosphorylation. Next we have an isomerase reaction where glucose, which is aldose phosphate, is converted to fructose, which is ketose. So this we call it as phosphohexose isomerase. So it is converting an aldose form into a ketose form. No change in the carbons. Then we have the regulatory step of the pathway. Always regulatory step will be shown with thick arrows because they control overall pathway. So this requires again ATP. It's catalyzed by phosphofructokinase. It's a regulatory step. ATP inhibits the step. AMP and fructose 2,6-bisphosphate will stimulate the step. The aldolase enzyme is acting on fructose 1,6-bisphosphate. It is breaking the fructose 1,6-bisphosphate to produce a ketose phosphate, which is dihydroxyacetone phosphate, and aldose phosphate, which is glyceraldehyde 3-phosphate. The dihydroxyacetone phosphate formed will be isomerized to glyceraldehyde 3-phosphate. And glyceraldehyde 3-phosphate is the one which will be taking part in the reactions. Of the 2 triose phosphates, glyceraldehyde 3-phosphate participates in most of the reaction. Dihydroxyacetone phosphates participates only in glycerol synthesis and then gluconeogenesis. So the 2 molecules of glyceraldehyde 3-phosphate which are reduced now subjected to oxidation step. This oxidation step requires NAD plus and inorganic phosphate. So the glycolysis enzyme which is regulated by inorganic phosphate is glyceraldehyde 3-phosphate dehydrogenase. So we get 2 NADH molecule and we get a high energy compound 1,3-bisphosphoglycerate. Once you got 1,3-bisphosphoglycerate, you can go for substrate level phosphorylation. So you give 2 ADP molecules and the 2 ADP will be converted to 2 ATP. The resultant compound is 3-phosphoglycerate. We call it as 3-PGA. So one substrate level phosphorylation reaction is over, catalyzed by phosphoglycerate or glycerokinase. Then the PGA molecule undergoes a mutation so that the phosphate group will be shifted to generate 2 molecules of 2-PGA. This is PGA mutase. A mutase is an enzyme that transfers a group from one carbon to another carbon. So from 3rd carbon, the phosphate group is transferred to 2nd carbon. This need arises because once 2-PGA is generated, it can be converted to high energy compound phosphoenol pyruvate. The sole purpose of having this step is that you convert a low energy compound to another low energy compound and finally convert the low energy compound into a high energy compound. So we have enolase reaction here. And as I said earlier, this reaction in clinical biochemistry is blocked by using fluoride. So we get 2 molecules of phosphoenol pyruvate, the second high energy compound or energy intermediate generated in glycolysis. Then this undergoes substrate level phosphorylation. The second substrate level phosphorylation. So we use 2 ADP, we get 2 ATP. And the product of the reaction is 2 molecules of pyruvate, which is 6 carbon. So this is the end product of normal glycolysis. So from here we have energy production phase. So you have shown the reactions, whichever physiologically irreversible as physiologically irreversible one, and whichever reversible one are shown as the reversible one. Then in the case of erythrocytes, we have a phenomenon called BPG shunt. This won't occur in the glycolysis of another cell, but it occurs in erythrocytes. So it will be taking place in the erythrocytes. It produces a regulatory molecule called 2,3-bisphosphoglycerate, which is negative allosteric regulator of oxygen binding to hemoglobin. So the BPG shunt begins at the level of 1,3-bisphosphoglycerate. So the dots indicate all other reaction up to that what you have here. Now in the normal one, what we have done is we made the 1,3-bisphosphoglycerate as a substrate level phosphorylation reaction. So we got ATP. In the case of erythrocytes, 1,3-bisphosphoglycerate will be mutated. So BPG mutase. A mutase will be shifting a group from one carbon to another carbon. So here from first carbon, the phosphate will be shifted to second carbon. And this goes for hemoglobin binding. Once it binds to the hemoglobin, hemoglobin ability to bind oxygen decreases, oxygen is released. Then this will be hydrolyzed, otherwise there won't be glycolysis. So this will be hydrolyzed, generating 3-phosphoglycerate in the rest of the glycolysis. So only this part is the one that's important for erythrocytes, because they produce 2,3-bisphosphoglycerate, which is a negative allosteric regulator of oxygen binding to hemoglobin, so that hemoglobin releases oxygen. When this pathway takes place, remember, there is no substrate level phosphorylation and therefore no ATP formation.